Welcome, everyone, and thank you for joining us virtually for the latest version of the Variant Effect seminar series. My name is Jan. I'm a member of the VEST organizing committee, along with Adrini de Souza, Diego Calderon, Ohana Bezerra, and Mariano Martin. These seminars are made possible with a lot of support and help from Lara Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effect Alliance. As a reminder, we're always on the lookout for potential speakers for future seminars. So if there is anyone you would like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have a few housekeeping items to mention. Each talk is around 20 to 25 minutes, but there should be time for a few questions. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you're a NAVE member, feel free to visit our seminar Slack channel for more discussion. And finally, we are posting updates on our Twitter account, so be sure to follow us there. You can catch the seminars from today and previous presentation that have been recorded on our affiliate CMAP YouTube channel. All the details of the seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. For the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. Today's first speaker is Anna Aksakova. She's a molecular genetics PhD candidate at the University of Toronto in the Roth Lab. The title of her presentation is Systematically and Proactively Testing Variant Effects for the Autoimmune Regulator Gene. Anna, whenever you're ready.
Thanks for the introduction. Let me get my slides up. You can see it?
Yes.
Great. Okay, so thank you so much for inviting me. I am really excited to share the work I've done towards the map for the autoimmune regulator gene. And what I wanted to start with was the variant interpretation crisis, which is familiar to many of us. But variants of uncertain significance are increasing over time in ClinVar, the database of genetic variants and their impact on human health. And this number is only increasing as more and more people get sequenced, but there's a need to evaluate these variants of uncertain significance to give more useful clinical interpretations, such as benign or pathogenic, that are useful for patients and clinicians. So to introduce the autoimmune regulator, which is a gene that a map would be useful for, this gene is involved in T cell development. And very briefly, the autoimmune regulator is important in the thymus. And so this is an autoimmune organ located between our lungs. And T cells, they originate from the bone marrow and they travel to the thymus, where this interaction of T cells and medullary thymic epithelial cells is really important for the transfer of information. And so if you think of the thymus as T cell university, this is where T cells learn what is around the body. And then they leave and they circulate around the body and they find their targets and pathogens and things like that. But what I wanted to focus on is this transfer information between the T cell and the thymic cell. And looking closer at this interaction, this is where the autoimmune regulator is important. And so in a normal situation where wild type AIR is present, AIR upregulates thousands of different tissue-specific antigens in the thymus. These get presented to T cells. And if the T cell can recognize that, it undergoes apoptosis. This is a checkpoint to eliminate self-reactive T cells. But if AIR isn't working properly, what happens is that the T cell that might be self-reactive doesn't see what's around the body and then goes out into the periphery and targets peripheral cells. And this leads to a condition called autoimmune polyendocrine syndrome type 1. So just a bit more detail about how it works. So it's widely debated whether it should be called a transcription factor. What we do know is that it regulates thousands of tissue-specific genes, but how it works is not completely understood. So I will go into these domains later when I show the map, but just to show you an overview, And yeah, so how it works is not by binding DNA likely. Instead, it's that it is targeting genes that already have hoist promoters. And in that way, it's interacting with a lot of different transcriptional machinery to upregulate genes. Okay, and so more about the disease that mutations in AIR can cause. This is a rare autosomal recessive disease called APS1, and sequencing is increasingly used to diagnose it. And there's a few reasons that it's useful to have knowledge about which variants cause disease before a patient develops severe diseases. It can be warranted to give immunosuppressive treatments to patients to prevent the progression of the disease. And you can appreciate how many different symptoms there are. There are over 20. And to be diagnosed with APS1, usually you have to have 2 out of 3 of these major symptoms in green. And 78% of AIR missense variants are currently annotated as variants of uncertain significance that are available in ClinVar. Okay, and so just to mention one of the symptoms, which is type 1 diabetes. So this is the symptom that I wanted to focus on in making my reporter, because autoantibodies to insulin are one of the ways that patients can develop type 1 diabetes. And so using this knowledge, what I wanted to do was make a reporter system that can use the insulin promoter sequence upstream of GFP. And for this, I'm using something that many of you are familiar with is the BXB1 landing pad cell line. And this cell line also has the flippin landing pad. And so every cell will have this reporter system integrated. And then AIR variants can integrate at 1 copy into each cell at the BXB1 landing pad site. Okay, so I developed this reporter system in the hex cells. I integrated this reporter system, and then I needed to evaluate whether the reporter worked. And so for this, I validated the reporter using a small set of clonal cell lines. And so these are benign and pathogenic variants that are annotated already in ClinVar. And so you can see that the GFP fluorescence in flow cytometry is shifted further to the right for the benign variants and the wild type compared to the pathogenic variants. And we don't need the separation to be perfect. We just need to be able to set a gate where we can select for more cells or enrich for the cells that are more functional and compare that to the rest of the population to pull out the functional variants. Okay, and so knowing that the reporter works, now I could do this in large scale. And so I generated a library in-house of almost all possible missense variants of AIR, which I used popcode for, a modified version of popcode, which some of you use as well. And so this works by oligos that have an NNN codon centered in the middle, hybridizing to the template strand to make all of the possible mutations. After that, the library gets large-scale cloned into a plasmid compatible with integration into the landing pad cell line. And here, when I sort the cells, I'm enriching for the top 10% using gates that I determined during my small-scale results. And sequencing this population versus the entire population allows us to generate a fitness landscape where we have all possible amino acid changes. Wild type residues are in yellow. We have variants that are dropped out in this population as more blue, so they're less functional. And red is an increased reporter expression function. So yeah, so there we go. So that's the protocol. And so in the actual output of the flow cytometry or the sorting, this is what the population looks like. So it's 1 peak, and I enriched for this top 10%. Okay, and so that leads me to the sequence function map for AIR. So we have good nonsense and synonymous separation, which is comforting. And the map, I will walk you through it. So this is the first domain. It's really deleterious. So in blue, we have it's the domain responsible for multimerization of AIR. And so this is really important for its function. And something else I should mention is that the last row is stop codons, so that should be blue as well. And we're picking up the nuclear localization signal, which is nice. There's a residue here that is interesting that is getting picked up as well. Moving along, we have a disordered region with some increased reporter expression variants that are pretty cool. We have the SAN domain. So this domain, it's been said that it binds DNA, but the residues that are supposed to be closest in proximity to the DNA are not the canonical sequence in AIR. And it is a little bit in the consensus track here, you can see that it is a little bit deleterious, but it might not be, this isn't definitively say whether it does anything, but it is interesting that there is a little bit of signal there. We can see that there's a patch here that the hydrophobic residues are deleterious in the SAN domain as well. Okay, and so the PhD1 domain, this domain interacts directly with unmethylated histone because AIR often upregulates, or mostly upregulates, genes in repressed chromatin environments, and this unmethylated histone is often found there. Okay, and so we're not capturing this domain as well as we'd hope, and that's probably because of the nature of the reporter. It's not in a repressed chromatin region, but we are picking up two residues here that I will come back to, as well as an increased expression here in one of the alpha helices, along with the second one here. So this is not completely looking like we'd expect, but it is picking up some interesting features. Next, we have the PhD2 domain, which is important for some functional aspects of AIR. It has a structure solved, which I will show you. But what's interesting is most of these residues are wild-type cysteines that are getting picked up as deleterious. And the C-terminal domain has been shown to be really important for AIR transactivation, and we're capturing that as well. So that's the full map. And so getting into more detail about what this shows us. So one interesting thing about AIR is that there is no solved structure. And you can see in the AlphaFold prediction here that there's a lot of spaghetti. So we thought that it would be cool to see if we would be able to pick up any interesting features by this, but what we're mostly seeing is that known structural regions are deleterious. So this is the card multimerization domain, which is the beginning of the protein that I've shown you. And there are some residues that are getting picked up that are not in the known structured areas, which I'm still looking into. But just zooming in on one of the domains where there are a lot of cysteines in the PhD2 domain, you can see that the residues that are closest in proximity to the cysteines are the most deleterious, as well as the larger sized amino acids. So if those are changed, the interaction with the zincs are not as solid. Okay. And here's the PhD1 domain, which is not completely captured by the map, but we can appreciate is that the residues around the zincs are when you mutate those, they increase the expression of the reporter. So that's an interesting finding we weren't expecting, and I'm still trying to figure out what that means for us, but it is pretty interesting, at least. Okay, and so then I wanted to follow up on some variants that got picked up by the map. And just to remind you, there is a patch of disorder that has increased reporter expression. And this is cool because there are no known gain-of-function AIR variants. And I was interested in seeing, are these real? Do they actually upregulate the reporter more than the wild type? As well as some new deleterious findings. So I pulled out some of the variants and I clonally tested them. And the follow-up variants do agree with map findings. And so the novel gain-of-function variants are increasing the reporter expression more than the wild type, which is super exciting. And these are the first reported gain-of-function AIR variants. Okay, and so using the map, we can now compare it to patient cohorts and see how well the map is doing, as well as how it compares to ClinVar. And so, oh, I'm not sure why the map is not, hasn't loaded properly, but I have done a precision recall analysis where we can evaluate how well is the map performing on a known reference set of variants. And I've compared this to computational predictors of variant effects. And so this is a balanced precision recall analysis where precision is the proportion of the map, the variants called damaging that are actually pathogenic, and recall is of all of the damaging variants, how many of them are actually of all the pathogenic variants, how many of them are actually damaging. And so what's important here is the further to the right the plot is going, the more the map is able to predict, or the predictor is able to, how well it's performing is shown by the crossing of the line through this precision at 90%. And so the numbers here indicate, so the recall at 90% precision, and the better one is the one that has a higher number. So anyway, what's happening is that Verity, one of the computational predictors developed in our lab, is working really well as well as Revel. And our map is performing on par with alpha missense, and ESM1B is also performing pretty well at evaluating error variance. What I do have to note is that the numbers here are lower. I wish I could see them, but the numbers are lower than we want because Revel doesn't show a prediction for all variants. So when you remove Revel, this gets better, or the map gets better because Revel removes a lot of the variant set that we have. So yeah, so that's the result. So at least the map is performing well. We know that it's capturing known variants. And just to show this in more detail, so here is our histograms of how the variants score depending on their categories. And so the benign variants are scoring higher than the pathogenic variants, which is good. These are conflicting ClinVar classifications, which are distributed. Quasi-benign variants are those that I've pulled from Nomad that are low frequency. And the variants of uncertain significance are leaning towards the benign, but there are some that are damaging as well, according to our map. Okay, and so next, what we can do with our map scores is provide evidence weighting using calculation. But first, what I wanted to show you was how do the ClinVar variants score on this histogram. And so each line is a benign variant or a pathogenic variant, and then missense variants are overlaid behind. And so using these histograms, what we can do is calculate a log-fold enrichment of pathogenic over benign, which means what is the likelihood that a variant is found is overlapping with the pathogenic distribution. So what this means is if you have a variant that is overlapping highly with the pathogenic distribution and you follow it down, it has a very high pathogenic, very strong evidence strength using this evaluation. And so with the new variants that we've now provided evidence for, we can see how many of them are moving according to this LLR score calculation. And the map provides evidence for most variants of uncertain significance that are currently in ClinVar for error. And so most of them are moving to benign, strong, benign supporting using map evidence, but some are going to patho support. And greatly overall, the known benign and likely pathogenic are going into the category that they should be. So this is good news. So at least the map has provided some evidence for variant interpretation. And next, what I wanted to do was look at how do map scores look when you look at a patient cohort. So what I did was looked at 12 APS1 related symptoms. So because there are over 20 symptoms, we prioritize some symptoms that are used for diagnosis or have a high concordance with disease. And what we did then was pulled out those patients that have low scoring rare variants and looked at how many of those patients have the symptom compared to the whole set. And then in the control, we did the same thing. And then I calculated the odds ratio between these two sets. And the only hit that we found that was significant was hypoparathyroidism. And Revel is also capturing this. But what this means is that there is a higher chance that people with the low scoring error variants have hypoparathyroidism than in general in the population. And also the ESM1B and alpha missense are not able to capture this significantly, but we are able to see that. And so just to show you more detail about what this table looks like. So there are 5 people with low scoring error variants and stop gain that have hypoparathyroidism. And in the set, there is 166 people with the rare low scoring error variants. And then calculating the odds ratio using the control participants, which is here, the odds ratio is 6.9 and the Fisher's p-value is significant. So we have a hit for the symptoms that is interesting as because this is one of also the diagnostic triads of APS1 diagnosis. Okay, so for future directions, because the PHD1 domain is not being captured as well as we'd hope, I would love to do CRISPR or RNA-seq to see if we can capture this this domain, just because the reporter system itself is limiting us to seeing that domain. Also, there's many different common alleles for error that one of them is at a prevalence of 24% in the population. So generating the map in this common allele background would be super interesting. Also, it would be cool to see if a different reporter for error function would give us the same result, because I've only used one promoter to evaluate error function. Because we think that error works on poised promoters and it probably doesn't bind DNA directly, this is probably, the insulin promoter is probably pretty good, but it would be really nice to confirm that this is showing the same thing for other reporters of error function, just because we only have one. And so in conclusion, this is the first error sequence function map, the first assay that can evaluate in high throughput. We know that this map performs well in the reference set and agrees with known predictions of variant effects. It also shows the first evidence of gain of function error variance. It has provided evidence for most variants of uncertain significance, and it has associated low scoring variants with hypoparathyroidism. Yeah, so I would really like to thank my entire lab for all their support and help, and I couldn't have done it without them. I also want to thank Annie at the sorting facility who helped me with all of the sorting, and our collaborators at the University of Bergen who are helping with the clinical side of things. So thank you very much, and happy to take questions.
Thank you, Anna, for the nice presentation. So now we are going to take the questions. So if you have any questions, please feel free to type it in the chat.
Oh, there's a question.
Yeah, we have a couple of questions here. So I am going to read first.
Okay.
What do you think is the mechanistic of GOF variant activity? Are only pathogenic variants correlated to the phenotypical features?
So I'm not sure what the mechanism is, but in the disordered region, the variants that are gain of function are they are hydrophobic, and there is no known structure in that area. So it's pretty difficult to say right now what the mechanism is. But what we do know is that there are no patients currently reported that have gain of function variants that I found. So if you have too much of the autoimmune regulator, it probably won't cause disease. But on a scientific level, it's interesting to find out what's going on. But the map can't tell us that, because it would be interesting to compare to the structure, but we're not sure. There is that patch of gain of function variants in an alpha helix in one of the domains, so that might be interesting and might increase binding to the zinc in that section. But I can't say right now what the mechanism is, but maybe someone can figure that out later with my map.
Great. And how could someone adapt DMS to test variants using closed chromatin?
Yeah, that's a really good, interesting question. Sorry, can you repeat the question? I can't.
Yeah. How could someone adapt DMS to test variants using closed chromatin?
Yeah, I think that if it's a gene that gets upregulated from closed chromatin, then there are ways out of that using RNA-seq and things like that. But it's hard to put a CRISPR reporter in a closed chromatin state. And I think I did try a CRISPR reporter, but what I think is happening is it's really hard to recombine into closed chromatin. So if we have a way to do that more efficiently, that would be great. So maybe if the efficiency was higher, it would be possible. But I think it depends on the gene that you're assaying. So for my case, I think that maybe single cell RNA sequencing with a lot of resources would be really interesting for at least that domain that I'm not capturing. But I would say it's really hard to do that. Yeah.
Okay. Thank you. I have a question. I'm curious because this gene that you are studying, you're studying at this moment, it is related with autoimmune. autoimmunity.
which?
With
Oh, yeah.
autoimmunity,
Mhm.
sorry. And I think maybe you have many resources from the literature about the mutations at this gene. And you showed us that your results are very accurate in terms of what is described before. And I would like to know if you have like a percentage number of how of your functional impact score are related with previous data banks or predictive models, and how those remaining percentage could be related with like systematic errors of your experimental approach.
So you're saying that the data about which patients have which symptoms are available, and to see if it agrees to the existing.
Yeah.
So one challenge in this project is that patients, even with the same variant, present different symptoms. Because the autoimmune regulator upregulates different genes in different people, it might not upregulate all of them. And so even patients with the same mutation will have completely variable phenotypes, even within the same family. So when people have looked at how the variants perform on phenotype, that's really challenging, and it hasn't been shown, an association hasn't been shown before that is clear between one variant and one symptom. But I think overall low scoring variants, the UK Biobank analysis is the best I can do, because that is, I can pull out all of those symptoms. But all of the patient cohorts, most of them have founder mutation populations that have the same variant. So the number of variants that I can access is the best in UK Biobank. Yeah, but I can look into more databases, but after some discussion, we think that this is the best way to approach it.
Okay, nice, yes. Let me see if we have more questions. Yeah, Drin is just saying thank you for the talk, and it will be super cool to find GOF variants that correlate features in UK Biobank. And just I have a last question. You were saying in your final slides what your aims for the future, but I would like to know more what kind of if you're thinking about like clinicians, how could your work help them like stratifying patients?
Yeah. So we use a model to predict. So when we calculate log likelihood ratio scores, which is sorry, right? So when we calculate these scores, what this does is convert the ACMG guidelines into the evidence strength. And so we're actually using the guidelines to give the variants these evidence strengths. And so if this contributes to the evidence strength classification by ACMG guidelines, we are helping interpret some of the variants, or at least provide evidence for them, especially the ones that we haven't seen before. So hopefully this in the future, this will be a useful resource for clinicians, but implementing this might be a challenge.
Yeah. Very interesting. Thank you so much, Anna.